So our topic for this meeting is alveolar ventilation. The ultimate importance of pulmonary ventilation is to continually renew the air in the gas exchange areas of the lung, where air is in proximity to pulmonary blood. So you can think of it as the goal of pulmonary ventilation is to replenish or to refresh the gases that are located in the alveoli, where the respiratory membrane is found and where gas exchange between the blood occurs. So you need to produce gross movement of air so that there is continuous uh, replenishment or refreshing of the air in the alveoli. The rate at which new air reaches this area, the alveoli, is called alveolar ventilation. So we have here a picture of the tracheobronchial tree. So at this level, you have the segmental bronchus, and that is the bronchus after the secondary bronchus. Remember that the tracheobronchial tree starts as the trachea and then it bifurcates into left and right primary bronchus, um, inserting or going inside the pulmonary hilum. And then at the level of the pulmonary hilum, the segmental bronchus results. So this is the segmental bronchus. And then the segmental bronchus progressively gives um, smaller branches or divisions called intrasegmental bronchi. Then that continues on to become small intrasegmental bronchi. And at this level, you already have 15 generations, meaning 15 splitting of the tracheobronchial tree. And then, once the cartilage are already absent and the tracheobronchial tree diameter is very small, you will have already encountered the bronchioles. So the bronchioles have uh, two particular types. You have a terminal bronchiole and you have a respiratory bronchiole. So at the level of the respiratory bronchiole, you will already see alveolar sacs embedded on the wall of the bronchiole. So here, even if it is also a conducting passage, you will already have gas exchange. And all of these will terminate in the alveolar sac and alveoli, which together, groups of alveoli, form an asinus. So, what is the reason why we are showing this picture? Because the pulmonary ventilation that we have discussed in the previous meetings, resulting from changes in intrapleural and alveolar pressure, factor in elasticity, the expansion of the lungs, and the movement of air causes air to travel along this tree and eventually refresh the air that is found here. Okay, and by constantly refreshing the air that is found here in the respiratory bronchioles and alveoli, one is able to facilitate gas exchange with each tidal breath or with each inhale and exhale, where 500 ml of air comes in and out of the lungs. The sinus, if it is enlarged, will look like this. So you have here the level of the terminal bronchiole and here the level of the respiratory bronchiole. you will see that the respiratory bronchiole is studded by alveolus. And the alveolus, as well as the bronchiole, are lined by elastic fiber, demarcated here as the green lines. And around this system, um, before the alveoli or alveolar sac, you will see smooth muscle encircling the tract. Okay, and then eventually it ends up in an alveolar sac where you will find a confluence of um, alveoli. You will see that each alveolus here has an opening connecting one alveolus to another, a small opening called the alveolar pore of Kohn. Okay, so that if one alveolus technically gets blocked, it can still communicate, although not as efficiently, through the alveolar pore of Kohn. Now let's talk about dead space. So dead space is the air a person breathes that never reaches the gas exchange area but simply fills the respiratory passages such as the nose, pharynx, and trachea. So for you to understand dead space, remember that the entire respiratory tract is not filled with alveoli. Only the terminal or end portions of the respiratory tree where there is alveoli conducts the process of air exchange. So that means the air that is present in the portions of the respiratory tree where there is no alveoli does not participate in air exchange or gas exchange. So, that portion that does not contribute to gas exchange is called dead space. On expiration, air in the dead space and not from the alveolus is expired first. So whenever the diaphragm relaxes and its elasticity causes it to ascend, and whenever the um, pull of the diaphragm is released, when the diaphragm relaxes, the elasticity of the lung due to the elastic fibers and the surface tension inside the alveolus causes it to collapse. What happens? Air moves because intra-alveolar pressure is higher than the environment. So that causes air to move out. However, what happens? When air moves out, the air that is in the dead space, meaning the proximal portions of the respiratory tree where there is no alveolus, goes out first, and the air from the alveolus goes out last. So during expiration, the dead space serves as a disadvantage for removing expiratory gas. Remember that the alveolus is the site where the air that you are moving in and out of the lungs comes in contact with the blood and where gas exchange occurs. So, during gas exchange, carbon dioxide from the blood moves into the alveolus. Okay? And that means the alveolus is now very rich in carbon dioxide coming from the cells of the body. So, if during expiration, the dead space is what moves out of the respiratory tree first, That means the gas that has accumulated in the alveolus, the carbon dioxide, is moved um, after only the dead space has been moved. So that means during expiration, uh, the gas that you want out of the body is moved only after the dead space is moved out. Okay, so that does not help the expulsion of carbon dioxide from the body. Okay, so you have here a picture showing to you the tracheobronchial tree of the right, as well as an endotracheal tube inserted. So this image, if you correlate it with the labels here in the right, will show you the location of dead space. Okay, so in here would be the total volume of exhaled gas. And you have here afterwards a color gradient. Okay, so um, first, remember that dead space is the volume of the respiratory tree filled by air that does not contribute to gas exchange because it has not come in contact with the alveolus. Okay, so first, this is the apparatus dead space where gas is found inside the endotracheal tube. And then you have here the anatomical dead space that is contributed by the pharynx, the larynx, and uh, even the tertiary bronchus before you encounter the alveolus. And furthermore, there are alveoli even if um, there is the presence of blood vessels around it, but those alveoli do not participate in gas exchange. So that is your alveolar dead space. Combine the anatomical dead space, which is the dead space produced by the absence of alveoli. Combine that with the alveolar dead space, which is the volume produced by alveoli that do not participate in gas exchange. So that means not all the alveoli participate in gas exchange. That's your alveolar dead space. Combine the two and you have the physiologic dead space. All of the alveoli that participate in gas exchange that contributes to alveolar ventilation, which is demarcated here as pink. So this is the procedure for the measurement of dead space volume. Okay, so you cause the patient to fully exhale, and then a patient suddenly takes a deep breath of pure oxygen which fills the entire dead space, the anatomic and the physiologic. Some oxygen mixes with the alveolar air but does not completely replace it. Remember, there is only replenishment of the air, not complete displacement of the air at the level of the alveolus. And that is because of the principle where as you increase the diameter of a tube, the movement of a fluid inside it slows down. Okay, again, as you increase the diameter or the cross-sectional area of a tube, the movement of fluid filling the tube slows down. So if we apply that to the tracheobronchial tree or respiratory tree, as more and more generations or divisions of the tracheobronchial tree results, what happens? You will produce smaller um, lumen uh, air passages. Okay, but because these air passages are more numerous, if you add up the cross-sectional area of all of these smaller passages, it will still result to a higher cross-sectional area. So that means as you go deeper into the tracheobronchial tree, the cross-sectional area, total cross-sectional area increases. And as you go deeper and as air moves deeper into the tracheobronchial tree, the flow of air, the speed, the velocity of air movement slows down. Okay? So that causes only some of the oxygen to mix with the alveolar air but does not completely replace it. Then what's next? All of that air um, that the patient takes in initially is pure oxygen. The dead space now is filled with pure oxygen at that point after inspiration. Then the person attaches his or her mouth through a nitrogen meter. Okay? And the person expires all of the air into the nitrogen meter. What then happens? The first portion of the expired air comes from the dead space, as what we have discussed a while ago, which is completely filled by oxygen. So, the volume of air occupying the dead space has a nitrogen concentration of 0. When the alveolar air begins to mix with the expired dead space air, nitrogen concentration in the reading rises rapidly. Why? Because the air that is in the alveolus is not completely replaced by oxygen, so it contains nitrogen. And when that air in the alveolus finally mixes with the dead space air, the nitrogen concentration will start to rise. After further expiration, there will be so much nitrogen recorded that it reaches a plateau, indicating that all of the air being expired at that time is already alveolar air. So this is better represented in this image. So what happens initially is the patient inspires pure oxygen, and then after fully filling the lungs, the patient starts to exhale. But remember that the oxygen occupies all of the alveolar dead space, all of the anatomic dead space. So the oxygen occupies all of the anatomic dead space. And what happens during expiration? You exhale the dead space first. So during exhalation, all of this gray shaded area is pure oxygen. Okay? And then, once you have already expired the anatomic dead space, the alveolar air now becomes expired. So, when the alveolar air is already being expired, remember that there is nitrogen in the alveolar air because it was not fully displaced by the inhaled pure oxygen. So when you start already expiring the alveolar air, the nitrogen concentration goes up until it reaches a plateau, and at this point of expiration, from 200 ml to 500 ml of expired air, that is already alveolar air. Okay? Meaning that is the air at the terminal or distal portions of the tracheobronchial tree. So, if you will observe this graph, you will see that there is a demarcation of the gray area and the pink area. Okay? And we can use this one to calculate or to determine the volume of anatomic dead space and the volume of alveolar air through this formula. The volume of dead space is equal to the gray area in the graph multiplied by the total volume of expired air. So this is in percentage. Okay? So percentage occupied by the gray area multiplied by the total volume of expired air. And the total volume of expired air is the entire space of the graph. So that will be divided by the percentage of pink area plus gray area, which will total 100%. Okay, so the percentage of the gray area times the total volume of expired air, which is 500 ml, divided by 1. So that will tell you the volume of dead space. Next, the normal dead space in a young adult man is 150 ml. So what does that mean? If the tidal volume or the amount of air being moved with regular inspiration and expiration is 500 ml, 150 ml of that is dead space air. What does that mean? Only 350 ml replenishes the alveolus and refreshes the air that is in the alveolus. Okay? So that the alveolus will have more oxygen for it to transfer into the blood, and the alveolus will have less carbon dioxide that comes from the blood after replenishment. Okay, so again, the 150 ml of the tidal volume does not participate in gas exchange. Why? Because it has no alveolus at that portion, okay, of the tracheobronchial tree. You have to take note that as you grow older, the volume of dead space air increases. So what does that mean? If you compare the dead space volume of a 20-year-old man and an 80-year-old man, the dead space volume in the 80-year-old man will be higher. Okay? So what does that mean? The amount of air that actually replenishes the alveolar air in an 80-year-old man, a very old man, is actually lower. Okay? And so they must have to breathe more air through an increase in ventilation so that they will be able to proceed with gas exchange. Okay, so that they can compensate for the increase in dead space air. So they have to breathe more. So we have encountered these two terms a while ago. Anatomical dead space is the volume of all the space in the respiratory tree other than the alveoli, where there is no gas exchange, because the portions of the respiratory tree where there is no alveoli does not participate in gas exchange. The volume of air present in that region of the respiratory tree is called anatomic dead space. No gas exchange because there is no alveoli yet. Okay, so that includes the nose, the pharynx, the larynx, the trachea, the primary, secondary, and tertiary bronchus, even the terminal bronchus. Only when you reach the respiratory bronchiole will you have gas exchange because there is already alveolus there. However, we have the concept of physiological dead space, when the dead space incorporates both the anatomical dead space and the dead space, meaning no participation in gas exchange, resulting from alveoli that does not participate in gas exchange due to poor blood flow in its capillaries. So what does that mean? In a patient, there will be alveoli that even if there is air inside, there is no gas exchange that occurs because there is no blood that surrounds the alveolus. Okay? So, that alveolus, even if there is air, because there is no blood, it does not participate in gas exchange. So, you have to incorporate the volume inside those alveolus as dead space. So again, physiologic dead space means anatomical dead space plus the dead space in the alveoli, air inside the alveoli where there is no blood flow. Okay, so that is your physiological dead space. So this is a good representation of dead space. So this is your simplified tracheobronchial tree. Because this portion still has no alveolus, the air filling this portion is your anatomical dead space. However, you have here depicted two alveoli. One alveolus has blood flowing around it, and so the air that is present here equilibrates with the blood. So there is gas exchange here. However, this other alveolus has no blood going around the alveolus. Okay? And so even if air is present here, there is no gas exchange because there is no equilibration with blood. And so you have to consider all of this as anatomic plus um, dead space inside the non-participating alveoli as functional dead space. So all of this is your functional dead space, even the alveolus where there is no gas exchange. In a healthy person, the anatomical dead space is equivalent to physiologic dead space. So the two are equal in a healthy person. Why? Because almost all alveoli are functional. Okay? So, whenever there is a healthy person, because all the alveoli contain air and all of the alveoli has blood flow in it, then the anatomical dead space and the physiologic dead space are equivalent. However, when there is pulmonary disease, where the alveoli are partially functional or non-functional, so that means there are alveoli that is filled with air but is not subserved with blood, no gas exchange. The physiologic dead space can be as high as 10 times the anatomic dead space. Because the normal anatomic dead space is 150 ml. In a diseased lung, especially in a patient with emphysema, the physiologic dead space, meaning anatomic plus the dead space in the non-functioning alveolus, can be as high as 1 to 2 liters. 10 times 150 means 1,500 liters, so around this volume. Next, we have the term alveolar ventilation. Alveolar ventilation per minute is the total volume of new air entering the alveoli and adjacent gas exchange areas each minute. So this is what I was telling you a while ago, that as the tidal breath occurs, there is a continuous replenishment of the air in the alveolus. And how does that replenishment occur? As air moves in and travels through the tracheobronchial tree, and as you go deeper in the tracheobronchial tree, the cross-sectional diameter of all of those air passages exponentially increases. And that will cause the air to flow slower and slower as you go deeper in the tracheobronchial tree. And at the level of the alveolus, um, the books, our physiology books, will tell us that there might not even be gross movement of air, but only equilibration of air. Okay? So, alveolar ventilation means the volume of new air, freshly breathed air, that enters the alveoli, replenishes the alveolar air, and causes gas exchange to proceed. So per minute, so you have a rate here. This is equal to the respiratory rate, how many breaths per minute you take, times the amount of new air that enters the gas exchange area with each breath. Remember, the definition here is new air that enters the alveolus. So what does that mean? That means the tidal volume, and you have to subtract the physiologic dead space from that. Okay? Because the dead space does not participate in gas exchange. The dead space, in particular the anatomic dead space, does not reach the alveolus. So you have to subtract that from your tidal volume. So that what you will have um, after removing the dead space would be only the new air that enters the gas exchange area. So your alveolar ventilation means respiratory rate, number of breaths per minute, multiplied by the difference between the tidal volume and the physiologic dead space, which in a healthy individual is 150. So let us say in a healthy individual who breathes 16 breaths per minute, you have here 16 times 16 multiplied by the tidal volume of usually 500 minus the dead space of 150. So that will give you 16 times 350. So this is equal to 5,600 ml. So what does that mean? Each minute, 5,600 ml of air reaches the alveolus and replenishes the alveolar air so that oxygen from the environment can continuously go into the blood and carbon dioxide from the blood can continuously leave the blood and go into the ambient air when it is expired. Alveolar ventilation is one of the major factors determining the concentrations of oxygen and carbon dioxide in the alveoli. Therefore, because of gas exchange, alveolar ventilation is one of the major factors determining arterial carbon dioxide and oxygen concentrations.